All right. Can we get going? Okay, so in case you missed the first part and didn't hear the announcement, I'm not Shin Aizawa from Kobe Riken, pretending to be him. I'm Utpal Banerjee from UCLA, and I'll be talking about signal transduction, stress response, and metabolic control of hematopoietic stem cells using Drosophila as a model system. Drosophila blood cells are of the myeloid kind. So normally one finds in the mammalian lineage both a lymphoid lineage which gives rise to T and B cells, etc., which are not present in Drosophila, but it's only the myeloid cell lineage which seems to have been ancient for the purpose of innate immune response, to which the other kinds of lineages have been added on later. There's also not an erythrocyte population. There's no need for carrying oxygen. The really nice thing and the take-home message from this talk will be that these cells are largely stress-sensing cells, and therefore, interestingly, they have used certain developmental mechanisms for their development, which are then also used for stress sensing. And this is the kind of finding that we're finding over and over again, enough number of times to believe that this is almost a general fact. And then we're hoping that this will also be true in mammalian systems. So this is for me to remind myself to tell you that the earliest progenitors that we have in Drosophila are sort of like the common myeloid progenitor in mammals. There isn't really a hematopoietic stem cell identified in Drosophila as a mouse person would call them. Now, the primary blood cell types, not so important to know their names. They're mostly macrophages called plasmatocytes. So these function as macrophages. They pick up dead tissue. They secrete antibacterial peptides and so on and so forth. There's some specialized cells called lamellocytes we don't need to worry about. Then there are these crystal cells. They contain crystals inside them of prophenol oxidase. This is the most similar to that will be the tyrosinase in mammals, which helps in wound healing, thickening, and it probably has other functions that are currently unknown. Now, when we started looking at the development of blood cells in Drosophila, basically what we knew was that the blood cells all come from these lobes of the lymph gland at the larval stages. And somehow, so this is stained with some marker which stains all of the different cells. And somehow from these bag of cells come all these different diversity of cell types, and it was completely unclear as to how these different cell types are generated. So Corey Evans, who is an amazingly observant postdoc in the lab, came back and said that he felt that this outside area looked spongy to him rather than the inside, and he wanted to call these two things two separate areas, this the medullary zone and this the cortical zone. And the reason why this made any sense at all to give them different names was because he had then co-stained with a differentiation marker, marker that stains differentiated or differentiating cell types. And what he found was that the differentiated cells lie in the outside cortical zone of the lymph gland, and this internal zone, which I will, as time goes on, you'll see corresponds to the progenitor class. They're not stem cells, but stem-like progenitor groups from which these cells arise. And then there will be a niche, hematopoietic niche, that we'll call the PSC in a minute. Now, the reason why these cells in the medullary zone are different, they've got different markers, they are functionally different. So these are little plastic beads, red-colored beads that have been put in, and these outside differentiating cells, being macrophages, they'll pick up these beads, whereas the stem-like progenitors, they will never pick up those beads. because they are still not differentiated yet. So unlike blood development in our bone marrow, which is really difficult to image and see, this is, of course, a very nice way to be able to look at what's going on. So I'll tell you several little short stories to make my point that both the differentiation pathways, which are very strange in these cases, and the stress-related pathways are kind of two sides of the same coin. First story is by Edward O. Suansa, who was a graduate student at the time. He made this remarkable discovery that the medullary zone cells, the progenitor class, have got high reactive oxygen species, in this case superoxide radicals. Now this was very strange because normally you would expect a stem-like population or a progenitor population to have very low levels of reactive oxygen. We all know they cause DNA damage and so on and so forth. Now this thing looks scaringly red, but actually it's not that high a level. This is physiologically generated ROS, which is kept at a certain high level, and then when the cell differentiates in the green cells, this ROS level is actually reduced down. So in mammals, in fact, this is not unheard of either. So in fact, in a paper from Gary Gilliland's lab, when we looked it up, all the hematopoietic stem cells have got low levels of ROS, which is true, but the myeloid progenitors, which is in fact what these cells are more like, have got 3 orders of magnitude higher ROS levels. And the explanation there was not quite clear, and what Gary and others and his lab basically said, well, these cells will at some point become phagocytes and maybe they need ROS, but that's not true because these are progenitors and they have no phagocytic function. The phagocytic function of ROS comes from non-mitochondrial sources. This is mitochondrially generated ROS. Now, in Drosophila, you can easily now see why these cells have high ROS. What you can do is you can reduce the amount of that ROS by, for example, expressing some kind of a scavenger protein which removes that ROS, or you can make a hypomorphic mutation in superoxide dismutase which will increase the ROS a little bit more. What you find is scavenging away that ROS causes lack of differentiation, and so the differentiation is blocked, the red in this case, whereas adding a little bit higher ROS from where the ROS was already causes extensive amounts of differentiation of this progenitor class. So the ROS has to be just right. This is one of those Goldilocks kind of a thing. It just has to be just right. So here, for example, we have now changed the ROS levels by an artificial means. We've taken a subunit of the mitochondrion, complex 1, and we have made a mutation in the complex 1 in those cells. What we find is it causes increased differentiation at a very early stage, where in wild type you'll never see differentiation, but you start seeing differentiating cells. And by the time wild type really shows differentiation, you see that when you mutate the mitochondrion, you get huge excess of differentiation in such circumstances. So, and this was linked to ROS by various genetic criteria. So again, increase in ROS will cause increased differentiation. Decrease in the ROS causes decrease in differentiation. This is when we realize that ROS at low levels, the physiologically generated levels, just like other noxious chemicals such as nitric oxide, carbon monoxide, etc., acts in this case as a signaling molecule. That signaling molecule is important for the maintenance of the progenitor fate, but increase or decrease will have an effect. Now this is good, and the reason is that under conditions of mitotic stress, then that ROS level can go further up, and that would then, we showed in those papers, that activates Jun kinase pathway, which activates through a FOXO and polycomb BMI in mammals dependent method, generates these lamellocytes. So it makes sense that a system which is at that point will result in mitotic stress, will cause it to differentiate into more and more immune type of cells. Now, so that was an example of how a cell needs ROS in order to differentiate, but then under stress conditions uses the same ROS signal in order to accentuate the differentiation and uses it as a stress signal and increases its differentiation potential and becomes a really useful example. Now, here's another example that I will talk about, which is the way we in fact got into this is when we started working on blood cells at all, was because we used to work on a gene called lozenge, which is a Runx protein, which actually Veronica also worked on. I have a paper with Veronica on this one protein, which I'm extremely proud of. The lozenge gene encodes what turned out to be a very important mammalian protein called Runx1. And the reason why it is important, Runx1, is because loss of Runx1 causes loss of all definitive hematopoiesis in mammals, and translocations into Runx1 locus leads to acute myeloid leukemia. So Tim Libesky, who was a very brave graduate student, tried to look to see if there was any blood defect in lozenge at all or not. And he's the one that introduced blood into our lab. And so at a non-permissive temperature of lozenge, you see loss of these crystal cells, and both in the larva and in the embryo. So lozenge is needed to make these crystal cells. So that was just an introduction to lozenge for a second, because I'll need that to explain the next point. But I wanted to introduce also this protein HIF, hypoxia-inducible factor. HIF in flies is called SEMA. And the reason why HIF is an important protein is normally under hypoxic condition, HIF is stabilized, goes to the nucleus, and turns on transcription. Under conditions of normal oxygen, HIF is degraded and is supposed to have no function whatsoever. And so it is that, hence it's called hypoxia-inducible transcription factor. However, Tina Mukherjee, a great postdoc in the lab, found that SEMA or HIF1 is actually stabilized in these crystal cells, those lozenge-positive cells, even under normoxic conditions. And what she found was that if you took away one copy of SEMA or one copy of the HIF1 locus, you cause a loss of these crystal cells. If you increased the level of SEMA genetically, you increase the amount of crystal cells. Now, we had known this to happen with one other thing. If you lose Notch, you lose crystal cells. If you activate more Notch, you get more crystal cells. Now, this kind of was, there was something in the back of our mind which had always been of interest to us. We had known that Notch is involved in crystal cell development, but something that we had never understood for a very long time, which is that these are circulating crystal cells, unlike the ones inside the lymph gland that I showed you. These circulating crystal cells, when you take away Notch very early, you don't see enough of them, so you lose them. But what we found was that even when you take away Notch after lozenge is made in them, meaning that they have already become crystal cells, they're already circulating, if you take away Notch in them, you lose those crystal cells. Now, Notch is one of those proteins which must, in the canonical way, bind to another membrane-bound ligand, such as serrate or jagged, delta, etc., in order to function. But these are circulating cells. They are not going and sitting somewhere, getting a Notch signal, going somewhere else and sitting, getting a Notch signal. And yet, they seem to be requiring Notch signal all the time for their maintenance. So looking at this a little more carefully, we found that the early Notch induction, if you lose that, then you get no crystal cells at all. So you, in fact, require Notch in a specification kind of manner for those particular blood cells. But late Notch induction, late Notch function, will cause a different phenotype, which is that in the absence of late Notch, you will cause these crystal cells to burst. So these crystal cells are like platelets, like when we have to get a clotting done, these cells burst and the little fragments are helpful in forming clots. Similarly, the crystal cell fragments release the crystals inside, and that helps in forming the actual wound healing. So this maintenance is very important, and that maintenance is now by the Notch protein. Now, I know nobody in the audience cares about crystal cells, nor do I. Okay, that's not the point. The Drosophila crystal cells are useless cells. I understand that. The point here is that there is a principle that is being developed here about notch function, and there's an early and a late function, and regardless of whichever cells they were, if these were T cells, your interest will perk up all of a sudden. Certainly Apurva's would. What is true is that it is this maintenance function by Notch which requires this hypoxia-inducible factor. So it is at this point that you need SEMA. So if it is at the specification point where Notch needs its ligand serrate, and it's at the maintenance point where Notch needs SEMA in order to maintain these cells. So if you were to take away the ligand early, you get no cells, and then later and later it becomes ligand independent. It doesn't require the ligand anymore. But on the other hand, with this late loss of SEMA gives you the same bursting phenotype, because you need that SEMA with a Notch later on in order to function together. So without going into a lot of details, this thing has been published now. Essentially what happens is, even under normoxic conditions, SEMA and Notch can bind one another. The fact that they can biochemically bind to each other intracellular domain was known in mammalian systems already. What this is showing is gives it an in vivo context for it. And this bound product is then involved in maintenance of crystal cells. Now, if you were to, so therefore this is a developmental pathway. Now, in terms of my point about the two sides of the same coin, where is this now used in, and I don't know if I put in the, no, I didn't. Where is this used for stress response? It is used for stress response because if you were to make the situation hypoxic, now you will get more SEMA stabilized because it is hypoxic situation. More SEMA will now bind more Notch, and under hypoxic condition, you'll get more crystal cells. So under that stressful situation, the fly will need more of this kind of a blood cell. So it is using this extremely weird developmental condition to make these cells, because under the stressful situation, it will use the same kind of a pathway in order to make more of this cell type. Okay, now the third little story. I told you about the two zones, the medullary zone that contains the progenitor class, and the cortical zone that we called it cortical because it was along the edge and it contains the differentiated cells. Now there's a third zone which we had called the posterior signaling center. Just about 32 or so cells, which we had found a long time ago, but didn't know what it really did at that time. But it expresses various kinds of signaling molecules. We called it the posterior signaling center. It expresses many signaling molecules, and it lacks the receptors for just about everything. Now, Lolitika Mondal, who is now at ICER Mahali, at that time did some experiments where she took away the PSC and by genetic means, and what she found was that there's a complete loss of the progenitor population and all the cells differentiate. So this is a typical example of a hematopoietic niche. A hematopoietic niche or microenvironment is needed for maintaining progenitor populations. If you were to take away the niche, it's not as if the progenitors die. It's just that they can't be maintained anymore, and in this case, they all differentiate into this ball of cells. If by genetic means she is able to increase the size of the hematopoietic niche, which can be done, now the number of progenitor cells goes up. And so this is, again, some old work that I'm just using as an introduction to what I'm going to say next. So this thing was known that there was a hematopoietic niche. And what Lolitika further showed that the hematopoietic niche contains the hedgehog molecule, and it is this hedgehog from the niche which, through some of these little projections, and we don't still understand how it reaches so far, is able to maintain the progenitor population in a progenitor way. If you were to lose hedgehog, this will become a ball of differentiated cells, just like when you lose the niche. So in this case, you have not lost the niche itself, but just removed hedgehog from the niche, and that then causes differentiation of the progenitor population. Now moving along to some newer stuff, what we found was that the one very important property of our progenitor population, perhaps the most important property of our progenitor population, is that they are quiescent. Many stem-like, progenitor-like populations are quiescent. They do not divide very much. They just hang out there, and they only divide when necessary. And what we found was, at some point, of course, all the cells divide a lot, and then they become quiescent. So they're receiving this niche signal. At some point of time, they become quiescent. And what we found was, this quiescence seems to match in time with when the differentiated cells are forming at the other end. So we proposed that perhaps these guys are sensing a signal back from the differentiated cell. This is a concept that there may be scattered ideas about this in mammalian systems, but there's no concrete example of this in mammalian systems, particularly not in hematopoiesis. We called this an equilibrium signal. The reviewer didn't allow us to call it equilibrium signal, but nevertheless, it's a backward signal, or whatever you want to call it. I'll just sneak in the word equilibrium signal here, hoping that the reviewer is not in the audience, without really meaning to suggest that it's some very complex equilibrium. What it is doing is, the idea is that between this signal and that signal, somehow we propose that it maintains progenitor here. So the idea is that if we either lose the niche signal, the progenitors will differentiate, or if we lose this backward signal from the differentiated cells, then the progenitor will also differentiate. And the simplest way to do this is if you kill some of the cells as they are differentiating, you don't get the backward signal, then these guys all differentiate. This was done as a very first test case, but killing cells is not a good way to do it, because that can send out distress signals and things like that. So we had to do it more properly genetically. And I do not have the time allotted to go through the entire genetics, which took a couple of years to complete. But after and couple of years and 5 postdocs to complete. So this is a couple of years, 5 postdocs worth of work, which then suggested that the way this works is a signaling molecule called PVF1, which is like the VEGF in mammals, is made in the PSC. It activates the PVR, which is the VEGF-like receptor in flies, PDGF VEGF receptor in flies, through STAT, activates a gene called ADGF, adenosine deaminase growth factor. Now, this was a great sort of a leap, which was made mainly because of mainly because Peter Bryant had said that Drosophila hemocytes require adenosine in order to divide. And Julian Martinez, who was an MD PhD postdoc at that time after his MD PhD, picked up on that and said, well, maybe these guys are dividing with, how many more? Okay. These guys are dividing because of adenosine. So he looked at ADGF, and adenosine is broken down by that ADGF, and then so these are the evidences. I won't go through it in detail. When you take away adenosine, ADGF, you cause full differentiation. If anyone is interested, I can go through the details of the genetics with you. Most importantly, the loss of PVR and STAT phenotypes, which are the ones which are sensed by the differentiated cells, are then rescued by overexpression of ADGF as the epistasis would require it to be. Now, the interesting thing here is, and if you want the details of the genetics, it actually, I haven't seen it yet, but it apparently came out last week in Cell. But the really interesting aspect of this thing is that the way adenosine works is by activating adenosine receptor. Adenosine receptor is a 7-transmembrane domain receptor, which works through the usual cyclic AMP pathway to activate pKa. What's really remarkable, of course, is that the hedgehog pathway inhibits pKa. And so the idea, this whole hypothesis that we had in our mind, that there will be a signal from hedgehog and there'll be a signal from the other end, without any proof whatsoever and without ever having done any biochemistry, it turns out that this is in fact exactly how this system works, which is that there is a balance of pKa which is created through adenosine, which is trying to increase the level of pKa, which would then cause division. Hedgehog is trying to block that, which will cause quiescence. ADGF is trying to remove this adenosine, which will increase that quiescence. So this is a beautiful system of having a signal from the niche and a signal from the differentiated cells, both trying to force quiescence onto a group of cells that are a progenitor cell class. One last little bit, and this is work by Ji-won Shim, who showed that in addition to these kinds of signals, which are inside the lymph gland, okay, is that the end of the, so I'm going to skip this. What she showed, that in addition to the signals which are inside the lymph gland, that there is also signals that come from outside. In particular, insulin from outside keeps these progenitors in an undifferentiated state. Amino acids keep these in an undifferentiated state. And so there's a whole signal between the fat body and the brain, which is involved in keeping the blood cells into an undifferentiated form. So it's not just a matter of the blood differentiating on its own, but signals from the brain and the fat body are also involved in keeping these blood progenitors in their own state. Okay, so I only talked about the work of the hematopoiesis, which is Corey Evans, Bama Mondal, and Tina, who worked on the adenosine project. G1 didn't get to at the end. And these are the recent past people. Julian Martinez, Lolitika is now here. Edward Osuansa is with Norway Perimon at Harvard. Sudeep is also at ISER Mohali. And Sergey has his own position as well. And the rest of my lab works on mitochondrial function. Thank you very much.
All right, beautiful talk. I have two questions, actually. So first one is picking on, I guess, the last slide, where these signals from the outside are coming in. And one thing that you show is wingless, and I was also thinking in the cortical zone versus the medullary zone, the first comment that you made in terms of the compactness and the diffused morphology. So is it anything to do with Wnt signaling perhaps stabilizing cadherins?
Yes.
And that, well, I mean, clearly, I mean, in many stem cell maintenance situations where Wnt have been involved. So that's one question. The second one is...
Let me answer that question because it's a complicated one and I'll forget it. Yes, wingless is involved. Yes, wingless is expressed in the medullary zone. Yes, wingless controls E-cadherin in the medullary zone. If you were to take away E-cadherin, those cells will lose their compactness and will start moving out. And as far as
they differentiate,
they will move out. Then whether they differentiate, that's a different signal altogether. It's not wingless related. If you now were to express wingless in the cells where it's supposed to be shut off in the differentiated, then they fail to differentiate. And in the insulin example, it is actually goes through the TOR pathway, which seems to be controlling, that wingless seems to be a target of that. So that's your first question, yes.
Thank you. The second one is I was curious about the SEMA, the HIF interaction with Notch, and at least in cancer situations, it's been shown that HIF can interact with beta-catenin and prevents its interaction with TCFs. So I was wondering if you could comment on that in terms of how perhaps nuclear signaling with HIF and beta-catenin maybe modulating.
I mean, that would be great. I mean, we have not looked at it. I mean, obviously, we have only looked at it from a developmental point of view. If it were to be interacting with beta-catenin, that would be another interesting example to look at here. We haven't seen it. Katie has a question.
Utpal, you showed pKa as the sort of node finally. So did you test pKa dominant negative or dominant active as well and see whether that would give the predicted functions?
Yes, I have. Yeah, all of that is there in the paper, and I also have it. As soon as you have some time, I would love to sit down and show you stuff. Yeah, it's also there in the paper. Yeah, we have used pKa, both the dominant negative. We've used the regulatory subunit and so on and so forth. So it all matches out. It's just really beautiful. Because there was one problem, one worry that we had was that whether or not all of this is because of hedgehog effect, and that you really did not need another signal at all. So that was an issue. The bell has rang, so I don't know if somebody had it. Manisha had a question, but maybe we can.
Comment on the ROS levels and differentiation with secondary probes?
Yes. Yeah, we have looked at it. Let me talk to you about that later. Let me check it out. Okay.
Utpal, can I ask one quick question? Do all the other signals also converge on pKa? All the other inputs to keep the progenitors in their undifferentiated state, do they also converge on the pKa activity?
I don't know how many more signals there might be that are these kinds of 7 transmembrane domain signals that are involved in this process. There is a unpaired domeless type signal which might be involved, but as far as we can tell, that's not a developmental signal. That signal comes in during immune challenge. So if there's an immune challenge, there might be another, because it's, as soon as you have a node, you can use that node to do anything you want.
That's what I was wondering, whether the insulin and the starvation
No, the insulin doesn't, because it goes through the AKT pathway and then it goes through TOR. So it goes through AKT-TOR pathway. That we have checked very nicely. That still does interact with the pathway, but it's unclear that that's a direct interaction. Yeah. Okay.